Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning, everyone. Today we will examine the vaccine development process with particular attention to clinical trials and approval procedures. I will guide you through preclinical work, the sequential phases of clinical testing, regulatory review and authorization, and finally post-approval monitoring and real-world evaluation. Throughout the lecture, I will use concrete examples to clarify each stage. We begin with preclinical research. Before any candidate reaches humans, investigators perform in vitro studies and animal experiments to assess immunogenicity and preliminary safety. These studies generate data on the antigen, the platform such as mRNA or viral vector, and the assays used to measure immune responses. Good preclinical data are necessary to obtain authorization for first-in-human trials. Clinical testing is typically organized in phases. Phase 1 involves a small number of healthy volunteers and focuses on safety and dose finding. Investigators collect data on adverse events and basic immunogenicity. Phase 2 expands the study population to hundreds of participants and refines dosing regimens and schedules. It also begins to assess immune correlates and generates information on the likely efficacy signal. Phase 3 is the pivotal stage. These are large randomized controlled trials with thousands to tens of thousands of participants. The primary endpoints in Phase 3 are clinical endpoints, such as prevention of symptomatic disease or prevention of severe disease and hospitalization. Regulators rely on robust Phase 3 efficacy data to evaluate risk-benefit profiles. In some modern programs, adaptive trial designs allow phases to overlap or to expand cohorts based on interim data. For example, seamless Phase 1-2 trials can accelerate dose selection. However, regulators still require rigorous evidence on safety and efficacy before granting full licensure. Regulatory review examines the totality of evidence. Agencies such as the FDA or EMA review clinical data, manufacturing quality and lot release testing, and stability data. During a public health emergency, regulators may issue emergency use authorizations that permit use based on interim data while requiring continued data collection. To illustrate, I will use the COVID-19 vaccine development experience. Several groups developed mRNA and adenoviral vector vaccines. Adaptive trials and accelerated review allowed emergency use authorization within months. Still, these programs included large Phase 3 trials demonstrating efficacy against symptomatic disease and robust safety monitoring. An important technical concept is the correlate of protection. This is an immunological marker such as a neutralizing antibody titer that predicts protection from clinical disease. Correlates allow regulators to consider bridging studies or immunobridging to extend approvals to new populations or to modified vaccine strains without repeating large Phase 3 trials. Manufacturing scale-up and quality control are integral to approval. Regulators evaluate process validation, potency assays, and cold chain requirements. A vaccine cannot be licensed without demonstrated capacity to produce consistent lots that meet release specifications. After licensure or emergency authorization, post-marketing surveillance begins. Phase 4 studies monitor rare adverse events and long-term effectiveness in diverse populations. passive surveillance systems, active safety surveillance, and pharmacoepidemiology studies all contribute data that can detect signals not apparent in clinical trials. We emphasize Phase 4 surveillance because rare adverse events may only appear after millions of doses are administered. This is why regulatory decisions often include requirements for continued follow-up, risk management plans, and public communication strategies. Finally, no single study answers all questions. The vaccine development process is iterative. Developers use immunogenicity data, effectiveness studies, and manufacturing controls to refine recommendations, update strains for evolving pathogens, and ensure that benefit outweighs risk over time. In summary, the vaccine development process moves from preclinical experiments to phase clinical trials, to regulatory review and authorization, and then to ongoing post-approval monitoring. Each step has specific aims and technical requirements, and together they protect public health while enabling timely access to effective vaccines.
Listen again to part of the lecture. Then answer the question.
We emphasize Phase 4 surveillance because rare adverse events may only appear after millions of doses are administered.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.